Hi, everybody. I wanted to go through applied learning assignment for Chapter 7 with you. Some of the questions seem to have caused some problems, and I just want to make sure we're going through some of the answers so you can understand how to come to the correct answers. So looking down the page, the first question we have here highlighted in yellow in the figure above, label each of the cells as being either diploid or being haploid. When a cell is diploid, it has both sets of chromosomes, meaning for any type of chromosome, it has one that had come from that individual's mother and one that has come from that individual's father. So these will be paired sets of chromosomes. In meiosis, what we're seeing here in this diagram, we're seeing one diploid cell being broken down or being divided into 4 separate haploid gametes. So kind of our first round of meiosis is shown by the first set of arrows. So within this round, you would have the prophase, metaphase, anaphase, telophase, and so forth. And then likewise in meiosis 2. So these little arrows are showing the entire process of the meiosis 2 divisions. So when we look at what, let me just grab a little tool here. When we look at this, the only cell here that is diploid is this first one. Okay, it has 2 chromosomes or a paired set. This first one is, let's say, the chromosome, the paternal chromosome. So here's the chromosome, and it's been duplicated, so it has its sister chromatid attached to it. The second chromosome would be the maternal chromosome, and we typically show these as blue and pink. So this would be the maternal chromosome that is homologous to the paternal one. So it's the same size, it's the same shape, it has the centromere in the same location. It codes for the same genes, but it may not be the same version of the gene. But here's the chromosome for mom, and then it is in duplicated state. So it has its sister chromatid attached to it. So even though we're seeing 4 chromatids, this is only 2 chromosomes. They've been paired up to be homologous to each other. So at this stage, the cell is diploid. From the end of the first round of division through to the end of the second round, all remaining stages here are haploid. Okay. In the diploid cell, question 1b, it says to circle and label the pair of homologous chromosomes. And I'm just going to grab another color to do that. The homologous chromosomes are all of this. So this is showing one set of homologous chromosomes. We would refer to this as a tetrad because they've been paired up. So this is going to be the homologous chromosomes. And as I said, they are paired in what we call a tetrad. To contrast this, the sister chromatids, let's grab another color here, are just 1/2 of the homologous pair. So these two are going to be the sister chromatids. And the sister chromatids are basically the 2/2 of the replicated chromosome. They are genetically identical to each other because they were formed in the process of DNA replication, and that process occurs in interphase S. So these sister chromatids were formed in interphase S. We can also see the sister chromatids are still connected to each other here at the beginning of meiosis 2, but they get pulled apart from each other in the second round. And so one of them ends up down in this cell, and one of them ends up down in this cell. Likewise over here, these two here are sister chromatids. Actually, let me fill this right in. Sister chromatids. They are basically, it's one chromosome, but you're seeing 2/2 of it there. And these are what then get passed down into the daughter cell. They will get pulled apart from each other in meiosis 2 to end up in separate daughter cells. Let's see what else it asks us to do next. Let me scroll down here. Oops, too far. Yes, it also wants us to label the gametes. So to This is the line of the metaphase plate. Okay, so in meiosis 1, we will see separation of the homologous chromosomes. In meiosis 2, we see separation of the sister chromatids. So that's going to be our answer to our next question down here. Meiosis 2 is going to separate the sister chromatids. Mitosis also separates sister chromatids because the homologous chromosomes, or those tetrads, don't form in mitosis. So the only thing that can be separated in mitosis are the sister chromatids. Remember, sister chromatids are identical to each other. So with one going into each of the daughter cells, that ensures that those daughter cells are identical to each other, and that is the whole goal of mitosis. All right. This final question generally causes a good number of errors. Looking at the figure above, what is the diploid number for this cell? So to give you a frame of reference, the diploid number for humans is 46, meaning in a cell that has a full complement of chromosomes, one from each parent, you will see a total of 46 chromosomes. To figure it out in this drawing, all we need to do is to count how many chromosomes are in our diploid cell. So here's our diploid cell up here, the first one, and we have 2 chromosomes. Here's 1 chromosome here and 1 chromosome here. Now they're in duplicated state, so we have 4 individual chromatids, but they are still only considered to be 2 chromosomes. They just happen to be in duplicated state. So the diploid number for the cell in our simulation is going to be 2. For the haploid number, we have to look at how many chromosomes are found in any one of the gametes. In humans, the answer would be 23. So this is the answer for humans. But in our little simulation we've got up here, we have only 1 chromosome in each of our gametes. And that makes sense because your haploid number is going to be half of your diploid number. So we have 2 chromosomes in our diploid cells, but only 1 in our haploid cells. All right, let's see what's on the next page here. The next thing we want to do here is to label the column that is mitosis, and that's this one. And that means this one over here is meiosis. How did I know that? Well, I know that mitosis takes 1 diploid cell and splits it into 2 genetically identical daughter cells, where meiosis will take a diploid cell and split it into 4 genetically different haploid cells. So easy to figure that one out. The next thing we want to know, what are some similarities in division between mitosis, cell division, and meiosis? There's a lot of different options here. One option is that the DNA is replicated before division. That happens in both of them. Both sets go through the familiar prophase, metaphase, anaphase, telophase stages. Just in meiosis, it goes through those stages twice. You're also seeing a process of cell replication or division. And there are other answers as well, but these are the typical ones that we see from students. The next table looks at this in a little bit more detail, though, looking at the differences between them. So here are our characteristics, and then on the other side, we want you to talk about them in mitosis versus meiosis. Most students don't have any problem with this one. Just double check you got your answers correct. The type of cells produced, mitosis is used for body cells or what we call somatic cells, whereas meiosis is used to produce gametes. We might also see this referred to as germline cells. Also, mitosis will produce diploid cells. So they are genetically identical to the original parent cells, where meiosis produces haploid cells. So either answer there is going to be appropriate. Number of daughter cells, as we saw up here, there are 2 daughter cells produced by mitosis, but there are 4 daughter cells produced by meiosis. Are the daughter cells genetically identical or different? This is important. In mitosis, they are going to be identical. Okay, the whole purpose through the process of, say, growth and development, those cells have got to contain the full genetic component, and it has to be an accurate genetic component. So it's essential that they be identical through the process of mitosis. Meiosis, however, being used for sexual reproduction, is producing gametes that are different from each other, or genetically different from each other. And this is important because it means that genetically the offspring are not identical to the parents. And that is important for genetic diversity in the population. And as we see in our lecture materials, it's important for the process of evolution as well. Final part of the chart here, the number of cell divisions. There is only 1 round of cell division in mitosis. But in meiosis, you can see we have 2 rounds of cell division. Okay, so again, most students are able to do that particular chart. However, this next diagram seems to have caused some problems this quarter. I want you to draw 4 chromosomes and show how they are lined up on the metaphase plate of both mitosis and meiosis 1. So in mitosis, I'm going to put my centrosomes here. This is going to be my spindle. So I'm basically going to have this as the line of my metaphase plate. So here I want to put 4 chromosomes in. So here's chromosome 1, but remember it's still in duplicated state at this point in time. So here we have 1, 2, 3, and 4 chromosomes all in duplicated state. All right, now how is this going to look for meiosis? Remember in meiosis, the homologous chromosomes will pair up with each other to form tetrads. So once again, here are my centrosomes, and here is my spindle. Here's chromosome number 1, just as I drew before. Okay, and it's in duplicated state, so it's got its sister chromatid attached to it. The chromosome 2, or the one that I should say is homologous to it, will have linked up with it to form these homologous pairs. And we end up with this structure that we refer to as a tetrad. So here are our first 2 chromosomes. Here's chromosome number 3 in duplicated state. And here's chromosome number 4 in duplicated state. Again, they have linked up with each other, the homologous chromosomes, to form what we refer to as a tetrad. This is the way you would see 4 chromosomes lined up in meiosis 1. All right, let's move on to our final page now. On this page, we're looking at how mistakes in meiosis can result in having an incorrect chromosome number in the resulting embryo. And we've used an example of Down syndrome for this. So here we have our question number 6. Suppose there's a mistake in meiosis. So a human egg receives 2 copies of chromosome 21. It should only have 1 copy in order to be haploid. But there's a mistake and it ends up with 2. So here is our egg, and here's, I'm not going to draw all the chromosomes, but here's the 2 copies of chromosome. This is number 21. Okay? It's fertilized by a normal sperm. So here's my little sperm, and here's his little flagella, and he has one chromosome inside of it. Remember, these will fuse on fertilization, and the nucleus of the zygote will get all of those chromosomes. So it will have 3 of these number 21 chromosomes in it. So our first question, how many copies of chromosome 21 would be in the resulting zygote? That answer is 3. Now, how many chromosomes or copies of chromosome 21 are going to be in each cell from the embryo that develops from this? Remember, it develops through the process of mitosis. So every single one of the cells is going to be genetically identical to the first one, which means every single cell is also going to end up with 3 copies of chromosome number 21 in it. All right, scrolling down, we'll finish up the last few questions on this page. We're showing you a karyotype, which is basically just a picture of all the human chromosomes that have kind of been sucked out of a cell and then laid out side by side and arranged so that the homologous pairs are laid out next to each other. So in this karyotype, we're asking you, is it taken from a cell that was in interphase, at the beginning of mitosis, such as prophase or metaphase, or at the end of mitosis in anaphase or telophase? And the answer here is B. It's at the beginning of mitosis. It's not in interphase because each of these chromosomes is in condensed state. Okay. If it was an interphase, it would be kind of in its long, stringy chromatin form, and we wouldn't be able to see them as individual discrete chromosomes. So we know it's not interphase. However, if we look at each and every single one of these chromosomes, here's a chromosome, here's a chromosome, here's a chromosome, here's a chromosome, you will see that each one of them is in duplicated state. Okay, it has its sister chromatid attached to it. That tells us that it's got to be at the beginning of mitosis in prophase or in metaphase, because in anaphase, those sister chromatids are going to get pulled apart from each other. So because they are still connected to each other at the centromere, we know it has got to be taken from either prophase or metaphase. So the answer here is B. We want you to label the sister chromatids in chromosome 3. And so here's chromosome 3. These two here are sister chromatids. And you could also label these ones here as being sister chromatids. And then finally, we want you to draw in that extra chromosome 21 to show if this was an individual with Down syndrome. So here's chromosome 21. We have 2 copies of it. All we need to do is just draw in a little third one there. The final question is one that students often think is complex, but it's deceptively simple. What do you think the reason is? So if you have an extra chromosome number 1, 2, 3, 4, or 5, it results in more severe abnormalities than having an extra chromosome 21. Why would that be? Well, let's just see what we can see. Here's chromosome 21. These are chromosomes 1 through 5. What's the most significant difference you see between them? They're bigger. And because they're bigger, they contain more genetic information. So there's more genetic information where you've got duplicate copies of it, and that can cause much more severe effects. So just a simple answer, it's more severe just simply because they're bigger and they contain more genes in them. All right, that's it. I hope that's helpful. Contact me if you have follow-up questions on any of that.